Welcome back from break. Today we're going to be re-looping the replication of DNA in our note packets as well as in our homework packets. So please take a moment right now and take out your DNA note packet. Turn to page 6 in your DNA packet. So we know that the process of DNA replication occurs within the nucleus, and it results in an exact copy of DNA. The original strand, which in this picture over here is shown in blue, is the template that guides the entire process. At the end of the DNA replication process, we result with two exact replicas of DNA, each of which have one parent strand and one daughter strand to make a conclusive double helix. At the result of DNA replication, there are 46 new chromosomes if we are replicating a human genome. The steps are quite simple. Step number 1, we unionize helicase at the origin. Helicase will unwind the helical structure, breaking the hydrogen bonds between the complementary base pairs and creating the replication fork. Helicase will continue to run down the double helical structure and opening up those hydrogen bonds in the helical structure. DNA polymerase is a very special enzyme. This enzyme is specialized at bringing complementary DNA nucleotides. Following the adenine to thymine, guanine to cytosine complementary base pair rules, DNA polymerase will build the new nucleotide strand. At the conclusion of DNA replication, we will have two exact copies of the original DNA. I'm going to take a moment. I'd like you to go ahead and pause the video. You're going to click this link right here. It's actually on the file as video number 1. Take a moment to go ahead and watch this video, review the content of DNA replication, and become a master at understanding the responsibilities of each enzyme within the process. Once you've completed that video, go ahead and come on back to the PowerPoint. Continuing further, now that you've had a pictorial representation of the process, we can see this is a great image showing us that the helical structure is being unwound and the result of the DNA replication process is two exact copies of DNA. Take a moment, pause the video right here, turn to page 7, and complete these DNA replication review questions. So as we can see, there's many enzymes involved, helicase being a very important one. DNA polymerase is also an important enzyme that unionizes the nucleotides together, building the complementary base pair rule. And ligase is also going to be an important enzyme, gluing certain portions of the DNA molecule together. Turn now to your homework packet. You're going to turn to page 3. This is a great little reloop. See if you can pause the video right here and fill in the blanks of the steps 1 through 4 of the process of DNA replication. So now that you've had a moment to go over these steps, let's go ahead and fill in the blanks together. Step number 1 is the unzipping process. We know that the enzyme helicase is the enzyme that's responsible for breaking those hydrogen bonds in between our DNA nucleotides. Step number 2 is the addition of nucleotides. This is done by DNA polymerase, and it's going to follow the complementary base pairing rules, A to T and G to C. Step number 3 is, like I mentioned, that zip-up molecule or enzyme called DNA ligase. Ligase is responsible for zipping that backbone up again in your DNA molecule and allowing for that helical structure to once again be reestablished. And the last step is the proofreading step. Proofreading is really quite important within the process of DNA replication to ensure that there are no mistakes or errors that are made throughout this process. And, DNA replication is a biological process that's important for the making of two DNA replicas. We know, just like seen in this image over here, if this is our original strand, that strand will be unzipped in this process by DNA helicase. And the parent strand, one of which will be going to the new strand and one which will be going to the old. And it will also result in having one daughter strand That is the new DNA strand. At the result of this process, we can see that there are two exact copies of DNA created. One which will then go into the new cell being created, and one that will go back into the old cell. Take a moment right here. We're going to practice some complementary base pair strand building. So take a moment, pause the video, and I'd like for you to take a second to build the complementary strands that pair with this strand A, and then B and C. Now that you've had a moment to build the complementary base pair strands, I'd like for you to check your strand. So strand A, we can see we're following the simple A to T and G to C. Take a moment, check your answer all the way through. Moving on to B, take a moment, feel free to pause the video to check your answers. And finally, strand C. Now, if your strands match mine, then you had no mutations in the process. And let's do one more practice together. If the sequence of nucleotides of one original DNA molecule were A, T, T, C, C, G, what would the new complementary replicated strand be? If you wrote down T, A, A, G, G, C, you are correct. However, in the process of replication, mutations actually do occur. Accuracy and repair, as well as proofreading, are essential in this process. And the process of DNA replication has a relatively high degree of accuracy. There's roughly about one error in every 10,000 paired nucleotides. However, if an error does occur, this error is referred to as a mutation. By definition, a mutation is any change in the nucleotide sequence in one or multiple locations along the DNA strand. Mutations can also be caused by chemicals, ultraviolet radiation. Often those chemicals are called carcinogens, and the combination of DNA proofreading is often done by DNA polymerase. Take a moment, pause the video. You're going to click video number 2 and watch the recording on cancer. This is a great introduction. Keep in mind as you are watching the video that you do have a Schoology quiz directly following the video. This will count as a quiz grade. There are 5 questions, each worth 2 points. If you'd like to rewatch the video more than once to make sure that you get your 100 on your quiz, that would be a wonderful idea. So now that you've watched the video and you've completed your quiz, which I hope you did really well on, we're going to keep moving on into mutations and giving you some specific examples. Take a moment now and turn to page 3 in your DNA homework packet. We're working on question number 2, good examples of mutations. So if you're a researcher at Harvard University and you had an original DNA sequence as shown below right here, and you had 3 mutated DNA sequences that are shown 1, 2, and 3 below. I want you to look at this first DNA mutated sequence and compare it to the original sequence. See if you can circle the individual nucleotide that has been mutated. Take a moment now, compare the strands, and circle the mutation. So when comparing the 2 strands, you probably circled the letter T, and that nucleotide is a good example of a substitution. We can see that the strand before is identical and the strand after is identical. This C has been replaced with a T, or substituted by a thymine instead of a cytosine. Take a second, look at strand number 2. Circle the mutation and see if you can predict what type of mutation it is. So examining those two strands, you probably circled the letter G. We can see that there has been a guanine inserted into the original sequence. Comparing the sequence before, we can see the TAC, and then we can compare the sequence following, and it's actually a continuation of the original strand. The variation here, or the mutation, is the addition of this guanine. This is an insertion mutation. Take a moment now, look at mutation sequence number 3, and identify the mutation. So you probably realize that there seems to be a letter missing between this guanine and this cytosine. And this is an example of a deletion. There has been a guanine that is missing from our sequence strand. This is referred to as a deletion mutation because a nucleotide has been removed or deleted. Take a moment now and watch video number 3. This is a re-loop of the replication process and all of the enzymes you will be held responsible for. This video has been a great recoup and re-loop of the entire process of DNA replication. Turn now to page 4 in your DNA homework packet. Take a moment here and complete question number 2, A, B, C, and D. Make sure that you're clear on your answers and you answers letter A through D in complete sentences. Once you have finished these questions, you can practice your knowledge by answering the multiple choice questions on the bottom of page 4. Once you've had some time to answer these questions, I'd like you now to go to Mrs. Shipley. She has a form for you called Breaking the Code. If you have time in class to go ahead and complete this, please do. Otherwise, this is expected that you finish it for homework. You are only completing the front side of the Breaking the Code ditto. You are completing the replication portion of this unit.